Available data related to bone specimen procurement for synchrotron microcomputed tomography remains sparse. Our comprehensive tutorial provides a methodology that is straightforward, minimally destructive, and cost-effective. Procuring bone specimens with consistent dimensions and a cylindrical shape is critical to ensure that the resulting data sets are of the highest quality and the results are replicable.
The techniques described in this manuscript are applicable to coring rocks, fossils, or any hard material. We've used these to collect uniformly sized cores of rocks and single crystals for high-pressure experiments to explore the rheology of Earth's crust and upper mantle.
Novice corers may find that the bone specimen forms conical shapes. This can be addressed by allowing adequate enough time for bone dust to flush from the drill bit and slowing down coring speed.
There's a lack of instructional text and videos that demonstrate bone specimen procurement for synchrotron microcomputed tomography and histological analyses. Our step-by-step bone preparation tutorial helps to fill this gap.
Place a 75 by 25 mm glass microscope slide on a hot plate set to 140 degrees Celsius and melt a generous amount of thermal epoxy resin on the center of the slide. Press the inferior aspect of the bone block into the thermal epoxy resin on the microscope slide with the length of the bone perpendicular to the slide. Shift the sample back and forth in order to coat the underside of the bone and ensure secure adhesion. Let the mounted specimen rest on the hot plate for approximately 5 minutes to allow the thermal epoxy to wick into the pores and cracks, making sure that the epoxy on the slide is free of bubbles. If bubbles are present, shift the sample back and forth to remove them.